The next session of random topics of physiology, human physiology in particular. So, schizophrenia, possible exaggerated function of the part of the dopamine system. Schizophrenia comes in varieties. One of the most common types is seen in the person who hears voices and has delusions, intense fear, or other types of feeling that are unreal. Many schizophrenics are highly paranoid with a sense of persecution from outside sources. They may develop incoherent speech, dissociation of ideas, and abnormal sequences of thought, and they are often withdrawn, sometimes with abnormal posture and even rigidity. There are reasons to believe that schizophrenia results from one of the most of three possibilities: multiple areas in the cerebral cortex, prefrontal lobes in which neural signals have become blocked, or where processing of the signals become dysfunctional because many synapses normally excited by the neurotransmitter glutamate lost their responsiveness to their transmitter. Then excessive excitement of a group of neurons that secrete dopamine in the behavioral centers of the brain, including the frontal lobes, and an abnormal function of the crucial part of the brain's limbic behavior control system centered around the hippocampus. The reason for believing that the prefrontal lobes are involved in schizophrenia is that a schizophrenia-like pattern of mental activity can be induced in monkeys by making multiple minute lesions in the widespread areas of the prefrontal lobes. Dopamine has been implicated as a possible cause of schizophrenia because schizophrenia-like symptoms develop in many patients with schizophrenia when they are treated with the drug called L-DOPA. This drug releases dopamine in the brain, which is advantageous for treating Parkinson's disease, but at the same time it depresses various portions of the prefrontal lobe and other related areas. It has been suggested that in persons with schizophrenia, excess dopamine is secreted by a group of dopamine secreting neurons. Those cell bodies lie in the ventral tegmentum of the mesencephalon, medial and superior to the substantia nigra. These neurons give rise to the so-called mesolimbic dopaminergic system that projects nerve fibers and dopamine secretion into the medial and anterior portions of the limbic system, especially into the hippocampus, amygdala, anterior caudate nucleus, and portions of the prefrontal lobes. All these areas are powerful behavioral control centers. And even more compelling reason for believing that schizophrenia might be caused by excessive production of dopamine is that many drugs that are effective in treating schizophrenia, such as chlorpromazine, haploperidol, and thioxazine, all either decrease secretion of dopamine at the dopaminergic nerve endings or decrease the effect of dopamine on the subsequent neurons. Firstly, possible involvement of hippocampus in schizophrenia was discovered when it was learnt that persons with schizophrenia, the hippocampus is often reduced in size, especially in the dominant hemisphere. Next topic is about thoughts, consciousness, and memory. Our most difficult problem in discussing consciousness, thoughts, memory, and learning is that we do not know the neural mechanisms of thought, and we know little about the mechanism of memory. We know that destruction of larger portions of cerebral cortex does not prevent a person from having thoughts, but it does reduce the depth of thoughts and also the degree of awareness of the surroundings. Each thought certainly involves simultaneous signals in many portions of the cerebral cortex, thalamus, limbic system, and reticular formation of the brainstem. Some basic thoughts probably depend almost entirely on lower centers. The thoughts of pain is probably a good example because electrical stimulation of human cortex seldom elicits anything more than mild pain, whereas stimulation of certain areas of hypothalamus, amygdala, and mesencephalon can cause great amount of pain. Conversely, a type of thought pattern that does require large involvement of cerebral cortex is that vision, because loss of the visceral cortex causes complete inability to perceive vision, form, or color. We may might formulate a provisional definition of a thought in terms of neural activity as follows. A thought results from pattern of stimulation of many parts of the nervous system at the same time, probably involving, most importantly, the cerebral cortex, thalamus, limbic system, and upper reticular formation of the brainstem. This theory is called holistic theory of thoughts. The stimulated areas of the limbic system, thalamus, and reticular formation are believed to determine the general nature of the thought, getting it such quantities as pleasure, displeasure, pain, discomfort, crude modalities of sensation, localization to gross areas of the body, and other general characteristics. However, specific stimulated areas of the cerebral cortex determine discrete characteristics of the thought, such as specific localization of sensations on the surface of body and other objects in the field of vision, feeling in the texture of skin, visual recognition of the rectangular pattern of a concrete block wall, and other individual characteristics that enter into one's overall awareness of a particular instinct. Consciousness can perhaps be described as our continuing stream of awareness of either our surroundings or our sequential thoughts. Then memory, roles of synaptic facilitation and synaptic inhibition. Memories are stored in the brain by changing the basic sensitivity of synaptic transmission between neurons as a result of previous neural activity. Changing basic sensitivity of synaptic transmission between neurons. The new or facilitated pathways are called memory traces. These are important because once the traces are established, they can be selectively activated by the thinking mind to reproduce memories. They are important because once the traces are established, they can be selectively activated by the thinking mind to reproduce the memories. Experiments in lower animals have demonstrated that memory traces can occur at all levels of the nervous system. Even spinal reflexes can change at least slightly in response to repetitive code activation, and thus reflex changes are part of the memory process. Also, long-term memories result from changed synaptic condition and lower brain centers. However, most memory that we associate with the intellectual process is based on memory traces in cerebral cortex. Positive and negative memory, sensitization or habituation of synaptic transmission. Although we often think of memories as being positive recollections of previous thoughts or experiences, probably the greater share of our memories is negative, not positive. That is, our brain is filled with sensory information from all our senses. If our minds attempted to remember all this information, the memory capacity of the brain would be rapidly exceeded. Fortunately, the brain has an capacity to ignore information that is of no consequence. This ability results from inhibition of the synaptic pathways from this type of information, and the resulting effect is called habituation, which is a type of negative memory. Conversely, for incoming information that causes important consequences such as pain or pleasure, the brain has a different automatic capability of enhancing and storing the memory traces, which is a positive memory. It results from the facilitation of the synaptic pathways, and the process is called a memory sensitization. As we discuss later, special senses in the basal limbic regions of the brain determine whether information is important or unimportant and make the subconscious decision about whether to store the thought as a sensitized memory trace or no, to suppress it. Classification of memories. We know that some memories last for only a few seconds, whereas others are last for hours, days, months, or years. For the purpose of discussing this type of memories, let us use a common classification that divides memories into short-term memory, which include memories that last for seconds or at least minutes unless they are converted into long-term memories. Intermediate long-term memories, which last for days to weeks but then fade away. And long-term memory, which once stored can be recalled up to years or even a lifetime later. So often we need to store our study materials like intermediate long-term memory, because short-term memory you will forget things after seconds or minutes, but intermediate long-term memory lasts for days to weeks. In addition to this general classification of memories, we also discussed earlier another type of memory called working memory, which includes mainly short-term memory that is used during the course of intellectual reasoning that is terminated at each stage of the problem is solved. Memories are frequently classified according to the type of information that is stored. One of the three classifications involved memory into declarative memory and skill memory. Declarative memory basically means memory of the variable details of an integrated thought, such as memory of an important experience that include memory of surroundings, memory of time relationships, memory of causes and experience, memory of the meaning of experience, and memory of one's deductions that were left in a person's mind. Skill memory is frequently associated with the motor activities of a person's body. Motor activities to extent like swimming, such as skills developed before hitting a tennis ball, including automatic memories into signing the ball, calculate the relationship and speed of a ball into the racket, reduced rapidly the motions of body and arms and racket required to hit the ball as directed, with all of the skills activated instantly based on previous learning of the game, and then moving on to the next stroke of the game while forgetting the details of the previous stroke. Okay. Then short-term memory. Short-term memory is typified by one's memory of 7 to 10 numerals in a telephone number or 7 other discrete facts for a few seconds to a few numbers in a time, but lasting only as long as the person continued to think about the numbers or facts. Many physiologists have suggested that this short-term memory is caused by continual neural activity resulting from nerve signals that travel around and around temporary membrane trace in a circuit of overperating neurons. It has not yet been possible to prove this theory. Another possible explanation for short-term memory is a presynaptic facilitational inhibition, which occurs in synapses that lie in terminal nerve fibers immediately before this fiber synapse with a subsequent neuron. The neurotransmitter chemicals secreted at its terminals frequently cause fertilization or inhibition lasting for seconds up to several minutes. Circuits of this type could lead to short-term memory. Then intermediate long-term memory. Intermediate long-term memory may last for several minutes to even weeks. They will eventually be lost unless the memory traces are activated enough to become more permanent. Then they are classified as long-term memories. Experiments in primate animals have demonstrated that memories of the intermediate long-term type can result from temporary chemical or physical changes or both, either in the synaptic presynaptic terminals of the synaptic postsynaptic membrane, changes that can persist for a few periods up to several weeks. These mechanisms are also important that reserve spatial description memory based on chemical changes in presynaptic terminals or postsynaptic neuronal membranes. Mechanism of memory studied typically by Kandel and his colleagues that can cause memories lasting from 3 minutes to 3 weeks in a large snail. Two synaptic terminals. One terminal, that is from a sensory input to neuron terminal directly in the surface of neuron that is to be stimulated, and it is called sensory terminal, and the terminal presynaptic ending that lies on the surface of the sensory terminal and is called facilitator terminal. And the sensory terminal is stimulated repeatedly but without stimulation of the fibrillar terminal. Signal transmission of the first is great, but it becomes less and less intense with the repeated stimulation until transmission almost ceases. Phenomenal habitation we learned earlier. So molecular mechanism of intermediate memory, mechanism for habitation. At the molecular level, the habitation effect in the sensory terminal results from progressive closure of the calcium channels to the terminal membrane, although the cause of this calcium channel closure is not fully known. Nevertheless, much smaller the normal amount of calcium ions can diffuse into the habitated terminal, and much less sensory terminal transmitter are therefore released because calcium entry is a principal stimulus for transmitter release. The mechanism of facilitation. In the case of facilitation, at least part of the molecular mechanism is believed to be as the following. Stimulation of the facilitator presynaptic terminal at the same time that the sensory terminal is stimulated causes serotonin release at the facilitator synapse on the surface of the sensory terminal. The serotonin acts as a serotonin receptor on the sensory terminal membrane, and these receptors activate the enzyme adenylyl cyclase inside the membrane. The adenylyl cyclase then causes the formation of cyclic adenosine monophosphate also inside the sensory presynaptic terminal. The CAMP activates a protein kinase that causes phosphorylation of protein that itself is a part of the potassium channel and the sensory synaptic terminal membrane. This in turn blocks the channels for potassium conductance. The blockage can last for minutes up to several weeks. Lack of potassium ions out of the terminal is necessary for rapid recovery from the action potential. The prolonged action potential causes prolonged activation of the calcium channels, allowing tremendous quantities of calcium ions to enter the sensory synaptic terminal. These calcium ions cause greatly increased transmitter release by the synapse, markedly facilitating synaptic transmission to the subsequent neuron. This, in a very indirect way, the associated effect of stimulating the facilitated terminal at the same time that the sensory terminal is stimulated causes a prolonged increase in excited sensitivity of the sensory terminal, which establishes the memory trace. Studies by Brain and colleagues also in the snail have suggested still another mechanism of synaptic memory. Stimuli from separate sources acting on a single neuron under appropriate conditions can cause long-term changes in membrane properties of the presynaptic neuron instead of the presynaptic neuronal membrane, but leading to essentially the same memory effects. Then long-term memory, no obvious demarcation exists between the more prolonged types of intermediate long-term memory and this true long-term memory. The distinction is one of the degree. However, long-term memory is generally believed to result from actual structural changes instead of the only chemical changes. So, structural changes are happening. These changes enhance or suppress signal conduction. Again, let us recall experiments of primitive animals where the nervous systems were much easier to study that are aided immediately by understanding possible mechanisms of long-term memory. Electron microscopy pictures taken from invertebrate animals have demonstrated multiple physical structural changes as many synapses during development of long-term memory traces. The structural changes will not occur if a drug is given that blocks protein synthesis in the presynaptic neuron, nor will the permanent memory trace develop. Therefore, it appears that development of true long-term memory depends on physically restructuring the synapses themselves in a way that changes their sensitivity of transmitting nerve signals. The following important structural changes occur: an increase in the vesicle release site for secretion of transmitter substance, an increase in the number of transmitter vesicles released, the increase in the number of presynaptic terminals, changes in the structure of the dendricular spines that permit transmission of the strongest signals. Thus, in several different ways, structural capability of synapses to transmit signals appear to increase during establishment of total to long-term memory traces. The number of neurons and their connection activities change significantly during learning. During the first few weeks, months, and perhaps even year or so life, many parts of the brain produce a great excess of neurons and send our neurons axon branches to make connections with other neurons. If the new way axon fails to connect with appropriate neurons, muscle cells, or gland cells, the new axons will diffuse within a few weeks. Thus, the number of neuronal connections is determined by specific nerve growth factors released from the stimulated cells. Furthermore, with insufficient connectivity occur, the entire neuron and the sending out of the axon branches might eventually disappear. Therefore, soon after birth, the principle of use it or lose it governs the final number of neurons and their connectivities in respective parts of human nervous system. It is a type of learning. For example, if one eye of a newborn animal is covered for many weeks after birth, neurons in that part of cerebral cortex, neurons normally connected to the covered eye will degenerate and the covered eye will remain either partially or totally blind for the remainder of life. Until recently, it was believed that every single learning is achieved in adult human beings and animals by modification of number of neurons in the memory circuits. However, recent research suggests that even adults use this mechanism at least to some extent. Consolidation of memory. In short-term memory to be converted into long-term memory that can be called weeks or years later, it must become consolidated. That is, short-term memory is activated repeatedly will initiate chemical, physical, and anatomical changes in the synapse that are responsible for the long-term type of memory. This process requires 5 to 10 minutes of minimal consolidation and 1 hour for strong consolidation. For instance, if a strong sensory impression is made in the brain but it's more followed by a minute or so by a electrically induced brain convulsion, the sensory experience will not be remembered. Likewise, brain concoction suddenly applicable to deep general anesthesia or any other effect that temporarily blocks the dynamic function of the brain can prevent consolidation. Consolidation and the time required for it to occur can possibly be explained by the phenomenon of rehearsal of the short-term memory as described. Rehearsal enhances the transferred short-term memory into long-term memory. Studies have shown that rehearsal of the same information again and again in the mind accelerates the potentials of the degree of transfer of short-term memory to long-term memory and therefore accelerates enhances consolidation. The brain has a natural tendency to rehearse newfound information, especially newfound information that catches the mind's attention. Therefore, over a period of time, the important features of sensory experiences become progressively more and more fixed in the memory stores. This phenomenon explains why a person can remember small amounts of information studied in depth far better than large amounts of information studied only superficially. It also explains why a person who is wide awake can consolidate memories far better than a person who is in a state of mental fatigue. Then, new memories are codified during consolidation. One of the most important features of consolidation is that new memories are modified into classes of information. During this process, similar types of memories are informations are pulled from the memory storage bins and used to help process the new information. The new and old compared for similarities and differences. The part of storage process store the information about the similarities and differences rather than to store the new information unprocessed. Thus, during consolidation, the new memories are not stored randomly in the brain but are stored in direct association with other memories at the same time. This process is necessary for one to be able to search for memory stored in date to find out required information. The hippocampus promotes storage of memories, and anterograde amnesia occurs after hippocampal lesions are sustained. Then some other aspects like retrograde amnesia, inability to recall memories from past. Hippocampus are not important in reflexive learning. Next topic, Alzheimer's disease. Alzheimer's disease, amyloid plaques and depressed memory. Alzheimer's disease is defined as a premature aging of the brain. Alzheimer's premature aging of the brain usually begins in mid-adult life and progresses rapidly to extremely lose the mental power similar to that seen in a very, very old age. The clinical features of Alzheimer's disease include the amnemic type of memory impairment, deterioration of language, vasospatial deficits, motor and sensory abnormalities, gait disturbances, and seizures are uncommon until the last phase of the disease. One consistent fading in Alzheimer's disease is loss of neurons in the part of limbic pathway that drives the memory process. Loss of this memory function is devastating. So Alzheimer's disease is a progressively and fatal neurodegenerative disorder that results in impairment of a person's ability to perform activities of daily living as well as a variety of neuropsychiatric symptoms and behavioral disturbances in the later stage of the disease. Patients with Alzheimer's disease usually require continuous care within a few years after the disease begins. Alzheimer's disease is a common form of dementia in elderly persons, and more than 5 million persons in the United States are estimated to be afflicted by this disorder. The percentage of persons with Alzheimer's disease approximately doubles with 85 years age, and about 1% of 60-year-olds and about 30% of 85 years old having the disease. Alzheimer's disease is associated with accumulation of brain beta amyloid peptide. Pathologically, one finds increased amounts of beta amyloid peptide in the brains of patients with Alzheimer's disease. The beta amyloid peptide accumulates in amyloid plaques, which range in diameter from 10 to several hundred micrometers and found in widespread areas of brain including the cerebral cortex, hippocampus, basal ganglia, thalamus, and even in cerebellum. The Alzheimer's disease appears to be a metabolic degenerative disease because of the accumulation of beta amyloid peptide. The key role for excess accumulation of beta amyloid peptide in the pathogenesis of Alzheimer's disease is listed by the following observations. All currently known mutations associated with Alzheimer's disease increase the production of beta amyloid peptide. Patients with the trisomy 21 or Down syndrome have these copies of the gene for amyloid precursor protein and develop neurological characteristics of Alzheimer's disease by midlife. Beta amyloid peptide. Then patients who have abnormality of a gene that control apolipoprotein E, blood protein that transport cholesterol to the tissue, have accelerated deposition of amyloid and greatly increased the risk of Alzheimer's disease. Then transgenic mice that overproduce the human amyloid precursor protein have learning the memory deficits in association with the accumulation of amyloid plaques, and generation of anti-amyloid antibiotics in humans with Alzheimer's disease appear to terminate the disease process. Then vascular disorders may contribute to progression of Alzheimer's disease. There is also accumulating evidence that cerebrovascular disease caused by hypertension and atherosclerosis may play a role in Alzheimer's disease. Cerebrovascular disease is the second most common cause of acquired cognitive impairment and dementia, and likely attributes to cognitive decline in persons with Alzheimer's disease. In fact, many of the common risk factors for cerebrovascular disease such as hypertension, diabetes, and hyperlipidemia are also recognized to greatly increase the risk of following Alzheimer's disease. Next topic, concept of dominant hemisphere. The general interpretive function of Wernicke's area and the angular gyrus, as well as for the functions of the speech and motor cortical areas, are usually much more highly developed in one cerebral hemisphere than the other. Therefore, this hemisphere is called the dominant hemisphere. What is that? interpretive functions of Wernicke's area and angular gyrus, as well as the function of speech and motor control areas, and usually much more highly developed in one cerebral hemisphere than in the other. Therefore, this hemisphere is called dominant hemisphere. About 95% of the people, the left hemisphere is the dominant one. Even at birth, the area of the cortex that will eventually become Wernicke's area is much as 50% larger in the left hemisphere than in the right, more than Even at birth, the area of the cortex that will eventually become Wernicke's area is as much as 50% larger in the left hemisphere than in the right in more than one half of neonates. Therefore, it is easy to understand why the left side of the brain might become dominant over the right side. However, if for some reason this left side area is damaged or removed in early childhood, the opposite side of the brain will usually develop dominant characteristics. The following theory can explain the capability of one hemisphere to dominate the other hemisphere. The attention of the mind seems to be directed to one principal thought at a time. Presumably, because the left posterior temporal lobe at birth is usually slightly larger than the right lobe, the left side normally begins to be used to a greater extent than in the right side. Thereafter, because of the tendency to direct one's attention to the better developed region, the rate of learning in the cerebral hemisphere that gains the first start increases rapidly, whereas in the opposite less used side, learning remains less well developed. Therefore, the left side normally becomes dominant over the right side. About 95% of all people, the left temporal lobe and angular gyrus become dominant, and the remaining 5% either both sides develop simultaneously to have dual function, or very rarely the right side alone become highly developed with full dominance. Promotor speech area, Broca area, located far laterally in the intermedial frontal lobe, is also almost always dominant on the left side of the brain. And the speech area is responsible for formation of words by exciting simultaneously the laryngeal muscles, respiratory muscles, and muscles of the mouth. The motor areas for controlling hands are also dominant in the left side of the brain in about 9 to 10%, thus causing right-handedness in most people. Although the interpretative areas of the temporal lobe and angular gyrus, as well as many motor areas, are usually highly developed in only left hemispheres, these areas receive sensory information from both hemispheres and are also capable of controlling motor activities in both hemispheres. For this purpose, they use mainly fiber pathways in the corpus callosum for communicating between the two hemispheres. This unitary cross-feeding organization prevents such interferences should create havoc with both mental thoughts and motor responses. A major share of our sensory experience is converted into its language equivalent before being stored in the sensory areas of the brain and therefore being processed for other intellectual purposes. When we read a book, we do not store the visual images of the printed words, but instead store the words themselves at their conveyed thoughts, often in language form. The sensory area of the dominant hemisphere for interpretation of language in Wernicke's area, and this area is closely associated with both the primary and secondary learning areas of the temporal lobe. This close relation probably results from the fact that the first introduction to language is by way of hearing. Later in life, when visual perception of language through the medium of reading develops, the visual information conveyed in the written words is presumably channeled through the angular gyrus and visual association area into the already developed Wernicke's language interpretation area of dominant temporal lobe is moved. When Wernicke's area in the dominant hemisphere of an adult person is destroyed, a person normally loses almost all intellectual functions associated with the language or verbal symbolism, such as ability to read, the ability to perform mathematical operations, and even the ability to think about logical problems. Many of such type of interpretive capabilities, some of much used in temporal lobe and angular gyrus regions of the opposite hemisphere, are retained. Psychological studies in patients with damage to the non-dominant hemisphere have suggested that this hemisphere may be especially important in understanding and interpreting music, non-verbal visual experiences, especially visual patterns, spatial relationship between one's person and surroundings, and significance of body language, intonations of people's voices, and probably any somatic experience related to use of limbs and hands. And even though we speak of dominant hemisphere, this dominant is primarily for language-based intellectual functions, and so-called non-dominant hemisphere might actually be dominant for some other types of intelligence. When you try to learn about body temperature regulation and cutaneous circulation, we could see portions like Normal body temperature, body core temperature and skin temperature. Normal core temperature. Then body temperature is controlled by balancing heat production and heat loss. Heat production, heat loss. Then insular system of the body. Some aspects related to cutaneous circulation. Blood flow to the skin from the body core provides heat transfer. Then control of heat conduction to the skin by the sympathetic nervous system. Basic physics of how heat is lost from the skin surface. Radiation causes heat loss in the form of infrared rays. And conductive heat loss occurs by direct contact with an object. Convective heat loss results from air movement. Cooling effect of wind. Conduction and convection of heat from a person suspended in water. Evaporation process. Evaporation is a necessary cooling mechanism at very high air temperature. Clothing reduces convective and convective heat loss. Sweating and its regulation by autonomic nervous system. Mechanism of sweat production. Then acclimatization of the sweating mechanism to heat, the role of aldosterone, other mechanisms of heat loss. Regulation of body temperature, role of hypothalamus, role of anterior hypothalamic preoptic area in thermostatic detection of temperature by receptor in the skin and deep body tissues. Posterior hypothalamus integrates the central and peripheral temperature sensory signals, neuronal effector mechanisms that decrease or increase body temperature. And some other aspects related to hypothalamus: stimulation of shivering, sympathetic chemical excitation of heat production, increased thyroxine production as a long-term cause of increased heat production. Then set point for temperature control, behavioral control of body temperature, local skin temperature reflexes, abnormal body temperature regulation in case of fever, resetting the hypothalamic temperature, regulation center in bile disease, effect of pyrogens, and mechanism of action of pyrogens in causing fever, role of cytokines, fever caused by brain lesions. Then some other aspects of characteristics of febrile conditions, chills. So crisis of flush, heat shock, harmful effects in high temperature, and acclimatization to heat, exposure of body to extreme cold, loss of temperature regulation at low temperature, frostbite, cold-induced vasodilation is a final protection against frostbite at utmost freezing temperature. These are the major points. We need to learn about the limbic system and behavior, activating driving systems of brain, control of cerebral activity by continuous excitatory signals from the brainstem, reticular excitatory area of the brainstem, excitation of the excitatory area by peripheral sensory signals. Then increased activity of the excitatory area caused by feedback signals returning from the cerebral cortex. The thalamus is a distribution center that controls activity in specific regions of the cortex. Reticular inhibitory area is located in the lower brainstem. The neurohormonal control of brain activity. Limbic system, functional anatomy of limbic system. Then reward and punishment function of the limbic system. Reward centers, punishment centers, association of rage and punishment centers. Plasticity and tameness. Importance of reward or punishment on behavior. Importance of reward or punishment in learning and memory, and habituation versus reinforcement. And specific functions of other parts of limbic system, like functions of the hippocampus, anterograde amnesia after bilateral removal of the hippocampi, theoretical function of the hippocampus in learning, functions of amygdala, effect of stimulating the amygdala, effect of bilateral ablation of amygdala, then overall function of the amygdala, function of the limbic cortex. Depression and manic depressive psychosis. So decreased activity of the norepinephrine and serotonin neurotransmitter systems. Schizophrenia we learned earlier. These are the major points related to limbic system.